So you want to identify variants in your sequence data. Let me help you. Variant calling can be quite challenging, especially for beginners. And that is why I'm making this video. And so by the end of this talk, you will have an overview of the variant calling process and what you can do after variants have been called or identified. Now with the variant calling, obviously you need to have your data. Now with the data, you can have FASTQ files, or you can also have your FASTA files. But for this tutorial, I will focus on the FASTQ format. So let's say you've had your sequences, your sequence data brought to you. You've had this from you've received your sequence data from the sequencing facility, FASTQ files. So they are the raw reads or the raw data. The first thing you have to do is to perform QC. That's very, very important. So with the QC is done because at the end of the day, you want results that are reproducible. You also want results that are of higher statistical power. And you can do that if you've done the QC. It's very, very important. So with the QC, there are two things that we look at here: inspecting the reads and also trimming the reads. Now let's look at the inspection. Now the inspection, we are looking at the quality. Okay, we are assessing the quality. So tools like FastQC can be used. FastQC will perform analysis and then also generate plots, and these plots you can look at them, you can visualize them. So with FastQC, it generates HTML files, and then the HTML files you can just open them using your browser, and then you can inspect the reads, okay, and assess the quality. So that is something that you can do. So once you have done that, then you can proceed. Once you do the QC, you know that the reads are okay, then you can proceed. Now let's look at the other QC, that is trimming. Now the trimming is done depending on the information that you have from the sequencing facility, and then the results from the inspection, the results from the FastQC reports, for example. So if, let's say, during the inspection you realize that some of the reads were of low quality, then you can use trimming to remove those reads. If, let's say, you were informed that there are adapters in the data, then you can use trimming to remove those adapters. You can also trim reads that are, let's say, short, or reads that in terms of length do not meet your threshold. You can do that using the trimming step. So with the trimming, tools like Genome, tools like Circle, and there are a lot of tools available that you can use for trimming. So just look at what works best for you and then use them. So that's about the trimming. So once you have done your trimming, you are ready to go. So let's proceed. Now your data has passed the QC stage. So the next thing you have to do is to map your reads to the reference genome. This means that you need to have your reference genome at hand. You need to have the reference genome beforehand. We normally use FASTA format. So you map your reads to the reference genome. And you should also note that the genome mapping process is computationally expensive. So you need to make sure you are using a high-end PC, or better still, use a computer server. Now with the genome mapping, tools like Browse Window Aligner, that is BWA, Bowtie, Minimap 2, etc. exists. So just look at the one that works best for you and then use that. During the mapping process, it's also important to set read groups. Some of the variant callers require that the BAM file that you supply have their read group sets. If you don't set the read groups, you may encounter problems or the tools may not work. So it's important to set the read group. So the read groups, when you set, you can input the sample ID, sample name, platform, etc. So again, if you look at the genome mapping tools, they have a way that you can use to set the read group. So just check them out. That's about it. Now, after performing the genome mapping, you have your alignment file, which will be the SAM format or the BAM format. SAM formats or BAM format, they are basically the same. First, they are basically the same. The main difference is that BAM files in terms of size are smaller, so they occupy less space. So we prefer to use the BAM first. So you have your BAM first. So there are a couple of things you also have to do here. One of the things that I advise you do is to inspect the BAM file. So you can use tools like IGV to do the inspection. So when you inspect, you'll be able to look at the reads, you'll be able to look at the mapping, you'll be able to look at the coverage and all that. And then you can also zoom into certain regions of the reference genome and then just inspect them as well. So we can do that using IGV, do the inspection, and that will help you to understand the data and also give you an idea of what to expect. Another thing you can do is to perform statistics. So we can look at, let's say, the number of reads that map to the reference genome. We can also look at the number of reads that did not map to the reference genome. We can also look at this in terms of percentage. So that will help you. Ideally, you expect that majority of your reads will map to the reference genome. And so once you do the statistics, you can use some tools to do that. Some tools, flag stats. Yeah, I think that's the option. You can use that to do the statistics. And then if, let's say, you do the statistics, you realize that quite a substantial number of the reads are not mapping or did not map to the reference genome, then you can inspect further. Okay, maybe you did not use the correct reference genome, or maybe there was contamination or there was a problem. The statistics will help you to look into that. So it's important to do that. Another thing you can do is to filter the BAM files. It's also important to do that. Now, with the variant callers, of course, they do some filtering and all that, but you can lessen the workload by doing some of these filtering activities before you start the variant calling itself. So the filtering, you can look at selecting reads that map to the reference genome. You can also look at maybe reads or maybe alignments that had a particular mapping quality. You can select them. You can use other criteria. So if, let's say, you are dealing with paired end reads, then you can also select reads that have their mate pairs also mapping to the reference genome. These are things you can do. So tools like SAM tools, BAM tools can help you do the filtering, and that will help you to get the quality data that you can use for the downstream analysis. So just make sure you do the filtering as well. So that's about it. So once you do the filtering, your files are getting ready. And so what you have to do next is to map duplicates and also remove them. In some cases, you can remove them if the software requires, but you need to mark the duplicates. Okay, that's important. Now with the marking and then removing of the duplicates, tools like Picard and some tools can help you do that. Of course, these tools have their strengths and weaknesses, so just make sure you read the documentation, and that will help you to decide which of these tools will be appropriate for you. So make sure you do that. So the marking of the duplicates is important because it will help you to avoid bias. If you don't mark your duplicates, you may introduce bias into your variant calling process, and that will affect the downstream analysis. That will affect the reliability of the result that you have. So you need to make sure you have marked the duplicates. And in fact, it's a requirement for some of these variant callers. So you need to make sure you have marked the duplicates, and in some cases also remove those duplicates. So you make sure you do that. So once you have that done, then you have the BAM ready files. So you can now proceed with the variant calling. So that's about it. Now with the variant calling, there are lots of tools available: GATK, FreeBias, VASCAN, etc. So there are a couple of tools available. Now before you call the variants, you need to consider these factors. I'll give 3 here, but there may be other factors you have to consider. The list I'll give here is non-exhaustive. One of the things that you have to consider is the ploidy of the organism. Are you dealing with haploids, diploids, triploids, etc.? This will help you to select the appropriate tool. I think for some of the tools, you can set what you call the ploidy. So they'll have a range of values that you have to use, and then you can use that to look at the correct sequence type. So that's something that you have to also factor in. You also need to look at the type of variants that you want to identify. Do you want to identify germline variants or somatic variant or structural variant, etc.? It's something you have to factor because some of the tools have been designed to identify certain types of variants. If let's say you are interested in germline variants, then tools like GATK, FreeBIAS, VCF tools can help you identify those variants. If let's say you want to identify somatic variants, you can use tools like MUTEC2 or VASCAN to help you identify those somatic variants. Now, if you are dealing with somatic variants, then the tool that you are going to use will likely require you to also have normal samples. I think MUTEC2 and then VASCAN, they will require you to have normal and then tumor samples as well. Now with MUTEC2, the developers have what you call the panel of normals. These are normal samples. Okay, so they were prepared using data from a 1,000 Genomes project, so you can use that. And you can also generate your own panel of normals. With VASCAN, you also need to supply your normal and then a tumor sample. So that's one thing you have to take note of. If let's say you want to look at structural variants, there are tools that can help you with that. GATK can help you identify structural variants as well. And then the type of variant will also determine what options or parameters to use when you are using these tools. So it's important to do that. Another thing you have to factor in is the sample size. Now let's take GATK for example. With GATK, the variant calling pipeline is almost the same for both small and then large sample sizes, but then along the way they change. So if you are dealing with a large sample size, aka large cohorts, then along the way the GATK documentation will tell you or tells you that this is what you have to do. These are the steps you have to follow. If you are dealing with a small cohorts, then this is what you have to do. So the sample size is very, very important here. So you take note of that. Let me also say that with variant calling, the process is computationally expensive, just like genome mapping, if not more. And so you need to have a high-end PC, or better still, use a computer server to identify the variants. So it's important to take note of that. So let's proceed. Now, let's say you have done the variant calling, you now have the results. With the tools for the variant calling, they will generate a VCF file format. The file format is VCF. So the file is called VCF. Let's just put it that way. So it's a VCF. Of course, some of the tools will generate other file formats. BCF tools, for example, can also generate .bcf files and then some other files as well. So that's what you need to. But the common one, the popular one, is VCF formats. So that is what I'll focus on now. Now, if you generate your VCF file, you are now ready to start the post-VCF analysis. But before you do that, it's important you do some filtering. You need to filter the VCF file so that you have high quality variants. It's important you do that. Now, the filtering, you need to decide the criteria you want to use. Let's say, for example, you can decide that you want to identify or you want to select variants that are present in this number of samples, or you want to identify variants that maybe let's say SNPs, or you want to use indels. That's also a criteria. Or let's say you want to identify variants that have this particular quality. There are also scores here in the VCF file. There are scores, and one of them is mapping quality. You can filter maybe based on mapping quality or other information. Let's say you also want to identify variants, or you want to select and use variants that were identified or that are present in a particular chromosome. You can also filter and then select those ones. So that's about it. There are a number of criteria you can use. So just look at what works best for you. You can read papers, look at what has been done, and also look at the best practices, and you can use that to filter and then select the variants that you want to use for the downstream analysis. You can also do some statistics, maybe look at the number of variants that were identified and also look at the number of variants identified per sample. Or you can also do some visualization. You can do some plots. BCF tools can help you to generate some plots. You can do some statistics. There are a number of things you can do here, and that also serves as a QC for you, and that will help you to make sure that things are in order. Okay, so that's important. So you can do that. It's a way of trying to get results that are reliable and reproducible, and results that are of high statistical power. So it's important to do all of these things. So let's proceed. Some tools also require that you normalize the VCF file. So normalization is done so that things are also, the files are in the correct format for some of these tools. So with the normalization, the VCF normalization, you can use tools like BCF tools to do that. So that is something you also have to factor. So depending on the tool that you want to use for a downstream analysis, you may need to normalize the VCF file. So take note of that. Now you've done everything, so now you now have a VCF file which is ready for analysis. Now the VCF file, depending on the tool that you use, you can have, if let's say you are dealing with, you have multiple samples, you can have your results in different files. If let's say you have 10 samples, you can have each of these, the variant identified for each sample stored in a particular file. So that means you end up with 10 files. Or some of the tools, you can configure them so that all of these will be aggregated into a single file. So that's something you also have to factor. So if the samples have been separated, then you can decide to merge all of them. You can use tools like BCF tools to help you do that. Or depend on your analysis, or depend on what you want to do, then you can also decide to maintain them in their separate files. So that is something that you also have to factor in. I think I've made one tutorial on how to merge VCF files, and there are also some for splitting VCF files. So you can just watch those ones as well if you have something of that sort to do. So that's something you have to factor in. Now we are ready. We now have our VCF ready file. So with the downstream analysis, there are lots of analysis you can do, but I will give a few here. Again, the list I will give is non-exhaustive. It's to serve as a guide for you. If let's say you are doing a population genomic studies, then you can look at computing FST values and then visualizing those values as Manhattan plots, and then you can use that to identify populations that are under selection. That's something you can do. You can also compute these values and then visualize as Manhattan plots and use that to make some inferences. If let's say you are dealing with tumor samples, then you can also do some annotation. In fact, the annotation here, annotation is actually recommended for all studies. It's important whether you are doing population genomic studies or whether you are doing some cancer study. I personally advise that you do the annotation, because annotation will give you information. Okay, information about the variants, and then you can use that to help you make certain inferences as well. So that's something you can do. So with the annotation, tools like SnipFF and Over can help you do that as well. And then you also have Gemini. I think Gemini is now being changed to Sliver. So that's another tool you can use to perform variance annotation or VCF annotation. So SnipFF can help you to identify the variant effects. So this variant that you have identified, what are their effects? SnipFF can help you get that information. And this information is also helpful if you are doing some cancer studies. Then you can use that to identify variant that's likely, or maybe there are possible causes of the cancer conditions that you are studying. So that's something you can also do. If you are dealing with GWAS studies, then you can use tools like P-Link to do your analysis and also make some inferences. So there are lots of things you can do here, but again, I'm only giving a few, so it's important that you take what I've given, use them as a guide, and use them as you see fit. So that's about it. So with the variant calling, I've already covered 2 tutorials, one on Snippy, which is another tool for variant calling. Snippy itself does not call the variant, but it uses a number of tools to call the variant. I've also done a video on BCF2, so you can just watch all these tutorials. I'll leave the links in the description box and also at the end of this video. And so if you also have anything to add, if you have some information which you think will be of benefit to others, make sure you put them in the comment section. Let's have a discussion and learn some new things. So that'll be all for this talk. Thanks for watching, and I'll see you in the next session. Goodbye.